folks. My name is Mark Benoit. I'm a third-year MD-PhD student at UConn. I made these videos on embryology of the nervous system to help better orient you to human neuroanatomy. I think embryology is very important for understanding neuroanatomy, and I think it will make it a lot easier for you. So I made it as quick as I could, and I hope you enjoy. Thanks. Okay, so there's this chart that a lot of textbooks like to show that explain where structures in the adult come from embryologically, and it's a terrible chart, and I'll explain why. So first I'm going to start with adult brain derivatives. This is easy to understand. You know where the cortex is, you know where the basal ganglia are, and then we move it to the thalamus, hypothalamus. They throw the retina in there to confuse you. Then they don't write midbrain, which is exactly what this is, and then they say superior and inferior nucleus, red nucleus, pons, the cerebellum's behind the pons, and then the medulla and the spinal cord. So it's basically a top-down view of the brain. Okay, then they show you these ventricles, and they try to, you might think that maybe they're part of these structures, but they're not. They're just kind of near them, and that's confusing. And then to make it even more confusing, they tell you the embryonic brain has different names, that like the prosencephalon, but then there's no structure that becomes the mesencephalon, and then they tell you the rhombencephalon becomes these two things. So the prosencephalon will split to become the telencephalon, diencephalon, mesencephalon stays that, and the rhombencephalon splits to the myelencephalon, and the spinal cord is always the spinal cord. So this chart is very confusing, and hopefully I'm going to walk you through it to explain it a little bit better, because it's important to understand neuroembryology to understand neuroanatomy in the adult. Okay, so we're going to talk about embryology and neurulations. So here's the embryo, and as you know, all the structures in the human body arise from three different layers. The ectoderm, which I've drawn in blue, gives rise to the skin and nervous system. The mesoderm, which I've drawn in red, gives rise to blood, connective tissue, spleen, bone, and it also contains the notochord, which is important for neuronal development. And the endoderm I've shown in brown, and that will form the gut tube and the lungs. So after 4 weeks, the embryo now looks like this, where the ectoderm completely surrounds the outside, but then there's also another ectodermal structure within that outer covering called the neural tube. The mesoderm still fills around on the inside, the notochord is still there, and the endoderm is still surrounded in the middle. Okay, but how do we get from the structure on the left to the structure on the right with the neural tube inside the outer covering of the ectoderm? Because we all know the nervous system is inside the skin. So halfway through, here's what the ectoderm looks like. The neural tube starts to invaginate as the outer covering extends around the outside of the mesoderm and the endoderm. And the reason it invaginates is because secreted factors from the notochord pull in the neural tube, and it eventually pinches off, so it ends up inside. And while it's closing, if I cut this neural tube and look down at it from above, you'll see that the neural tube closes first in the middle as it grows both caudally and rostrally. So here's the neural tube. Here are two openings. You can see it closed first in the middle as it grows rostral and caudal. And to me, this looks like a cannoli, so I call this cannoli stage. And eventually, both ends fuse. One part, the rostral part, is at the head, and the caudal part is at the end of the spinal cord. And let's imagine if I now take a slice through this tube, like this, after it's closed. And since the tube is closed, it's going to be a sealed chamber that looks like this. And when neuroanatomists first looked at this structure, they saw three bumps. And so the tissue that surrounds the outside, they figured, should have three different names. So the tissue that surrounds the first bump, they called the prosencephalon, which means forebrain. Second bump they call the mesencephalon, which is midbrain, and the third rhombencephalon, which is hindbrain. And that chamber that's in the middle will eventually become filled with CSF. So they call this the three-vesicle stage. So the three-vesicle stage later on in development becomes the five-vesicle stage, which all that means is that the three bumps that they saw before now look like 5 bumps. And I've numbered them, and they also gave them new names. And so the tissue that was the prosencephalon gets split into 2 different bumps instead of just 1 bump. And so they called them 2 new names, which is telencephalon and diencephalon. Forget that arrow. So the number 1 I drew before, that was that 1 vesicle. Well, now it's become 2 vesicles, which I call 1A and 1B. And don't be confused that 1A shows up twice because since it's symmetric, they called them the same. Mesencephalon bump stays the mesencephalon bump. So that's why it retains the name mesencephalon. The rhombencephalon tissue becomes 2 bumps, and so they named them metencephalon and myelencephalon. So that third chamber becomes 2, which I call 3A and 3B. And the rest of the tissue here becomes the spinal cord. And mesencephalon, metencephalon, myelencephalon, you can remember in that order because it's alphabetical. Mes, met, and my. Okay, so each one of these bumps will become tissue in the adults. The telencephalon becomes the cortex. The diencephalon becomes the thalamus, retina, and lens. That's why the little chunk is separated. Mesencephalon becomes the midbrain. Metencephalon becomes the pons and cerebellum. And the myelencephalon becomes the medulla. The rest of the tissue will become the spinal cord. Quickly, I'd like to submit a gripe that something that makes me upset about neuroanatomy is that everything has 3 names, and a lot of times people refer to the thalamus in the adult as the diencephalon, and it's not the diencephalon, it's the thalamus, because the diencephalon is an embryologic structure that becomes the thalamus, retina, lens, hypothalamus, and a few other structures. And you'll see sometimes someone will point to the thalamus in the adult brain and tell you that it's the diencephalon. And that's just something you have to get used to. I don't believe it's true, but that's the way the world is. Okay, moving on. So what are the inner chambers inside this tube going to become? They will become the ventricles in the adult. These 2 1A structures will become the 2 lateral ventricles, which technically are ventricle 1 and 2. It's just that left could be 1 or right could be 1, and that's why they didn't call them 1 and 2, and that's why it jumps to third. So it's lateral, lateral, then third. Then the second chamber here will become the cerebral aqueduct, which passes through the mesencephalon. That's why the cerebral aqueduct is in the midbrain. And then the fourth ventricle is kind of a combination of these 3A and 3B chambers, which is why the fourth ventricle hangs out around the pons, cerebellum, and medulla. Okay, if I took the tube at this stage and I made a cut and I looked down the barrel of this tube, so remember rostral is up and caudal is down, then it would look like this. So the tissue is around the outside and then the chamber is in the middle. And so rostral is coming out of the page. And around the ventricle are stem cells. So they call this the ventricular proliferative zone. And these stem cells have these processes that extend all the way to the outside of the tube. And so these are called radial glia stem cells because they make this radial pattern, I think. So the stem cell will divide and form a neuron. And the neuron in its embryologic stage crawls up the stalk until it reaches the outside. So all these stem cells are producing neurons, and they're all climbing up all these stalks, and they're all accumulating around the perimeter of this structure. And eventually, there are so many cells that accumulate around the outside that there's not enough surface area for all the cells. So it starts to fold and invaginate like this. And if you're wondering why the brain looks like the way it does, it's because of this concept. So here's the gray matter on the outside. Here are all the cell bodies. And you can see that the tissue is folded because of this concept. And so here's the ventricle. This is where the proliferative zone was. And the neurons would crawl all the way out to the outside and accumulate along the perimeter. And so they'll hang out here, and then they'll extend an axon either across the corpus callosum to the other side of the cortex, or they'll extend their axon down the internal capsule into the spinal cord. And I know what you're thinking. You're thinking, well, what about these gray matter structures? These aren't part of the cortex. They're not along the outside. And these are basal nuclei, which are involved in movement and other things that you'll learn about. And these are sort of structures originating from lower species. That's the way I think of it. And so they don't form the same way the cortex does. Obviously, embryology is more complicated than I'm making it out to be. Okay, I hope that was helpful for you, and there'll be more videos to come that will further expand on these concepts that we learned today. Okay, thanks.